The researchers claim that the drug is one of a kind because it is made to protect insulin-making beta cells in the pancreas from being attacked by the body's own immune system cells. According to the researchers, the drug's specificity for such cells may make it possible to use it long-term in humans with few side effects. Monoclonal antibodies are made by cloning, or making indistinguishable imitations of a creature, counting human, cell line. The discoveries, detailed online as of late and in the May issue of Diabetes, raise the chance of a new medication for type 1 diabetes, an immune system condition that affects around 2 million American kids and grown-ups and has no fix or method for anticipation. Dissimilar to type 2 diabetes, in which the pancreas makes too little insulin, in type 1 diabetes, the pancreas makes no insulin in light of the fact that the insusceptible framework goes after the pancreatic cells that make it. The absence of insulin obstructs the body's capacity to manage glucose levels. According to Dax Fu, PhD, the research team's leader and associate professor of physiology at the Johns Hopkins University School of Medicine, if the condition is not managed properly, people with type 1 diabetes face lifelong injections of insulin and many complications, including stroke and eyesight problems. According to Fu, MABF3 binds to a small protein on the surface of beta cells, which cluster together to form islets. The drug was made to protect beta cells from immune system cells that attack them as invaders by acting as a shield or cloak. The scientists utilized a mouse rendition of the monoclonal immunizer and should foster a refined rendition for concentrates on in individuals. A weekly intravenous injection of MABF3 was administered to 64 non-obese mice bred to develop type 1 diabetes for the current study at 10 weeks of age. All of the mice were healthy at 35 weeks. One of the mice created diabetes for a while, yet it recuperated at 35 weeks, and that mouse had early indications of diabetes before the counteracting agent was regulated. The researchers administered weekly doses of MABF3 to 5 of the same type of diabetes-prone mice until they were 14 weeks old, after which they continued to administer doses and monitor them for up to 75 weeks. According to the researchers, no adverse events were found, though one of the group's 5 members developed diabetes. Compared to the control group of mice that did not receive the drug and lived approximately 18 to 40 weeks, the mice in the experiments in which MABF3 was administered early on survived for the entire monitoring period of 75 weeks. Then, the specialists, including postdoctoral colleagues Devi Kasanathan and Zheng Guo, looked all the more intently at the mice that got MABR3 and utilized a natural marker called Ki67 to check whether beta cells were duplicating in the pancreas. They said, after treatment with the immunizer, safe. Cells withdrew from beta cells, diminishing how much irritation nearby. Furthermore, beta cells gradually started recreating. MABF43 in blend with insulin treatment can possibly progressively lessen insulin use while beta cells recover, eventually taking out the need to utilize insulin supplementation. For glycemic control, says Katsanathan, the exploration group found that MABF43 explicitly bound to beta cells, which make up around 1%, or then again 2% of pancreas cells. One more monoclonal counteracting agent drug, teplizumab, was supported by the U.S. Food and Medication Organization in 2022. Teplizumab ties to lymphocytes, making them less hurtful to insulin-producing beta cells. The medication has been displayed to defer the beginning of clinical stage 3 type 1 diabetes by around 2 years, giving small kids who get the infection time to develop and figure out how to oversee deep-rooted insulin infusions and dietary limitations. It's conceivable that MABF3 could be utilized for longer than teplizumab and postpone diabetes beginning for a significantly longer time, possibly however long it's controlled, says Fu. According to Guo, we aim to develop a humanized version of the antibody and conduct clinical trials to test its ability to prevent type 1 diabetes and to learn whether it has any off-target side effects. This is a continuous endeavor that the company intends to pursue, Different researchers who added to the examination incorporate Dylan Sarver, G. William Wong, and Maria Golson from Johns Hopkins, Shumi Yun from the College of Maryland, Aaron Michaels. What's more, Liping Yu from the College of Colorado. Also, Shandan Sona and Matthew Poi from Johns Hopkins All Kids Medical Clinic.